Okay, we're going to start on Chapter 2 this week, the chemistry of life. So having a general understanding of chemistry is necessary to understand a lot of the processes of life. So we're going to spend some time in Chapter 2 introducing and reviewing some of the basic chemistry terms and concepts. Then we're going to look at how they apply to some of the fundamental biological processes. This is a very full chapter. There's a lot of information here, and we're going to skim a lot of concepts. So we're starting on page 30, and we're going to start with the most general definitions. First of all, in chemistry, a substance is a form of matter that has characteristic properties. Now, an atom is the smallest stable unit of all matter, and it's comprised of three particles: protons, neutrons, and electrons. Protons and neutrons are found in the nucleus, while electrons move around the nucleus very quickly. And they move around in what can be described as an electron cloud made of different energy levels. The number of protons is what tells us what kind of an element the atom is. Now, protons have a positive charge and electrons have a negative charge. An atom in its pure state will always have the same number of electrons as protons. An element is a pure substance composed of a unique type of atom. And they're given names and placed on the periodic table, and we use the symbols from the periodic table to abbreviate the names of the elements. Those symbols are not abbreviations, they're symbols, but we use them in place of the element name. Now, a molecule is a group of atoms that form a unit with specific chemical properties. In a molecule, the atoms may be the same element or different, and we use a small subscript with the element symbol to show how many atoms of the element there are in a molecule. Now, the numbers next to the letters that you see here on the slide, they should be smaller. They should be a subscript, but for whatever reason, they did not translate onto the slide that way. So these compounds is the next one, and compounds are molecules that contain two or more atoms from different types of elements, and they're held together by chemical bonds. So all compounds are a type of molecule, but not all molecules are compounds in this context. So let's focus on atoms for a minute. I mentioned that electrons have different energy levels, and each energy level can only hold a certain number of electrons. This is the octet rule that you'll learn about in chemistry. So depending on the number of electrons in the outer level, which are called valence electrons, you often end up with an energy level that isn't full. So basically, and this is just a simple explanation, when an atom that has a spare electron or two or more comes near another atom that has an open spot for another electron or two, they try to combine or pair up. And they can do this in a couple of different ways. One way is that an atom will sometimes gain or lose an electron. And when this happens, the atom becomes charged. And charged particles are called ions. Cations, and I'm emphasizing the cat, and you'll find out in a minute, cations are positively charged, which means they lose an electron. Anions are negatively charged, which means they gain an electron, because electrons are negatively charged. A way to remember this is cats have paws. So cat ions are positive. This behavior of the electrons, where you can gain or lose one to become an ion, is why we use the number of protons to determine an element. So how do charged particles interact with each other? What is our general rule in science? Opposite charges attract, like charges repel. So when two oppositely charged ions come together, they form an ionic bond. Now look at the bottom of page 31. Here you have a sodium atom that has one extra electron in their outer level, and the chlorine that has a spot for one. So sodium becomes positively charged, and chlorine becomes negatively charged. So they are then attracted to each other and become ionically bonded, because sodium has given away an electron and chlorine has taken that electron, and then they're attracted to each other and become bonded. Sometimes electrons are just shared instead of moving to another atom. And when two atoms share an electron, they have a covalent bond. In this case, it's the pull on that shared electron from the nucleus of each atom that keeps the atoms together instead of the attraction of the ions or atoms themselves to each other. Sometimes they don't share equally. Water is an example of this. The hydrogen and oxygen molecules in water don't share their electrons evenly. Oxygen is a little bit of an electron hog. This type of bond can happen with a couple of other elements as well, but we're really going to focus on oxygen. Because the oxygen molecule gets more time with the electron than the hydrogen, it has a hint or a slight negative charge. And this means that the hydrogen ends up with a slight positive charge. We call this polarity, like the poles of a magnet. And water, this means that water is made up of polar molecules. This creates a weak pull between the oxygen molecules of one water molecule or the oxygen atoms of one molecule and the hydrogen atom of another. This weak pull creates a bond between these atoms, and we call this type of bond a hydrogen bond. Hydrogen bonds are weak bonds that involve the interaction of a hydrogen atom in one molecule to an oxygen atom in another molecule. This can happen with fluorine and nitrogen also, just as a note, just so you know. So take a moment and look at figure 2.4 on page 32 in your book. This illustrates the hydrogen bonds between the separate molecules of water. Okay, so looking at the slide, I want to make sure we can notice the differences between covalent and hydrogen bonds. Covalent is a strong bond between two atoms in the same molecule. Hydrogen bonds are weak bonds between atoms in different molecules. That's an important distinction. So this unique type of hydrogen bond is responsible for so many of the properties of water. And water is so essential to life that our book spends an entire section discussing these properties. This starts on page 33 in your book, so make sure you're there. On this slide, you'll notice I have the properties of water listed just in a simple list, but this is the same as what we're going to go over in your book. This is just notes to help you keep it straight. So because water is polar, it can dissolve lots of things. Both polar and ionic substances have charges. Because of this, their molecules can become attracted to each other. That means they can dissolve into each other. So molecules of polar substances are either attracted to the opposite poles of their own molecules or the opposite poles of other polar or ionic molecules. If a substance doesn't have polarity, we call it non-polar. So this is important. Non-polar solvents can dissolve other non-polar substances. Like dissolves like. Polar and ionic dissolve polar or ionic substances because they both have charged particles. Okay? So take a minute to look at figure 2.5 at the bottom of page 33. This illustrates what happens when salt dissolves into water. So table sugar or sucrose is a polar molecule, so it dissolves like this into water. The water molecules arrange themselves so that their partially negatively charged oxygens are pointing towards the positively charged sodium ion. Okay, and the partially positively charged hydrogen atoms are pointed towards the negatively charged chloride ion. And the water molecules are arranged so that the oxygen atoms in one molecule is close to the hydrogen atom from another molecule. And all of this is caused by the water's polarity. So look at that figure again and see how they're all arranged there. So molecules like salt that have a tendency to be attracted to water are called hydrophilic. Those are the polar and ionic. Molecules that have a tendency to repel water, kind of like oil, are called hydrophobic. These are the non-polar molecules. Or non-polar molecules. So it is this hydrogen bonding that keeps water molecules close enough to each other to keep it in a liquid state at room temperature. So we're going over the third property of water listed in your box here. Boiling water will add enough energy to make the molecules move farther apart. All the other molecules that are chemically similar to water, in your book list a few of those. These are gases at room temperature. They don't take as much energy or heat to change to gas. The difference is that they don't have an oxygen molecule or an oxygen atom. Hydrogen bonding occurs between molecules with hydrogen and oxygen atoms. So hydrogen bonding also creates another important property of water that makes it unique. When water molecules are in liquid form, they have more freedom to be pulled closer together, and it makes water molecules, the water, denser in liquid form than when it's in solid. If it didn't have this property, life couldn't exist in many parts of our world. Lakes freeze top first, which allows aquatic life to survive at the bottom and become insulated from the cold. Look at the graph. You can see when water is a liquid, those molecules can all get closer together, kind of intertwine. When it's an ice, those attraction hydrogen bonds become more rigid and there's more space between them, so it becomes less dense. All right, the next property of water that we're going to go over is cohesion and adhesion. Hydrogen bonds also mean that water molecules exhibit cohesion. Due to their attraction to each other, these molecules have a tendency to stay together even when exposed to tension. So cohesion on the surface of a liquid is called surface tension. Look at the image on the slide there. You can see a water strider is able to be on top of water because of the cohesive forces between the molecules. Water droplets are also formed because of the cohesive forces between those molecules. They're pulling towards each other, and that's what creates this water drop, this rounded water drop. Adhesion is a property in which the molecules are attracted to other substances. So adhesion of water allows it to adhere or stick to other surfaces. This is how it's able to make the surface of a structure or substance wet or keep it damp. So water's cohesion to itself and adhesion to the surfaces of other substances, it is what allows water to travel upwards in plants. Without this, there wouldn't be any tall trees or large plants. We'd only have small mosses or similarly sized plants. The molecules adhere to the sides and are pushed up, and as the molecules go up, they pull the other molecules that they're attracted to with them, like a chain. This is what we call capillary action. So another aspect of water that we kind of touched on already is its high heat capacity, which also gives it a high temperature of vaporization. The hydrogen bonds between the molecules of water means it takes a lot of heat energy to get the molecules to move. And to separate. This means that it takes a lot of energy to raise its temperature and get it to boil or turn into water vapor. Other liquids like oil or alcohol boil or vaporize at a much lower temperature. Now, being liquid at room temperature, like we said before, is very good for organisms. It's good for us. Being able to take a high heat is good for us. It means we won't dry out very quickly. And our temperature doesn't change as quickly since we're made of a lot of water. So we're able to handle a larger temperature range. All right. So we are going to move on to section 2.3. So the chemicals in our world can generally be divided into two groups, organic and inorganic. There is, of course, some overlap because our world is an interdependent system. So organic chemicals consist of the molecules that are essential for life. And when we study biochemistry or organic chemistry, these are the chemicals that we are going to study. We are on page 38 now. Organic molecules have a few key characteristics. So look at page 38, the second paragraph, and underline or highlight these characteristics. First, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur are the only elements used for nearly all organic molecules. Second, in organic molecules, they usually arrange themselves in specific or special groupings that we have labeled functional groups. And these groups interact with each other. Third, individual organic molecules join to form long chains called polymers. Now, the word polymer just means many parts. It's a Greek word. Okay, we are going to focus on the 4 specific classes of macromolecules or macros that are very important to life: carbohydrates, lipids, proteins, and nucleic acids. We're going to start with carbohydrates. Look at the definition of carbohydrate on page 38. Underline it and highlight it. It's a molecule composed of carbon, hydrogen, and oxygen, in which there are twice as many hydrogen atoms as oxygen atoms. So what is the ratio of hydrogen to oxygen? 2 to 1. There's a 2 to 1 ratio of hydrogen to oxygen. This is very important. What other molecule do we know of that has twice as many hydrogen atoms as oxygen? Water, H2O. So carbohydrate means watered carbon. Okay, so that's very important to remember. Carbohydrates are found in all living tissue. They are formed by green plants from carbon dioxide and water during photosynthesis. If you look on the slide, I just have an illustration there, the photosynthesis reaction. And as you see on the right-hand side, we have glucose and oxygen. And on the left-hand side is the carbon dioxide and water. We will study this more in depth later in the book, but I just want you to have an exposure to it right here. At the top of page 39, you'll see the hexagon shape, which is the most common one found in nature of a carbohydrate, and the way that you'll see it most often represented in organic chemistry. Now on the slide, I basically have the same figure, but this one actually has the letters, the symbols of the elements in it, so you can kind of get a better understanding of it. So all carbohydrates are made up of one of these hexagon or units of sugars, one of these hexagon shapes here. And we call this a saccharide. So carbohydrates can be classified by how many saccharide units are joined together. This one hexagon structure represents one sugar molecule or a monosaccharide. Okay, so one saccharide is called a monosaccharide. If you have a substance with just one saccharide, it's a monosaccharide. We call this particular one glucose. Another type of monosaccharide is fructose. Now these are simple sugars, and they taste sweet to us because their simple shape fits more completely and interacts better with our taste receptors for sweetness. So sometimes these join together and form disaccharides, which also can taste sweet. Page 39, figure 2.9 shows an illustration of that happening. You have a glucose and a fructose that join together to make a disaccharide, sucrose, and a water. Okay, so that's very important here. When glucose and fructose come together, there are two hydroxyl groups. That's the OH groups there that you see illustrated on the saccharide. They react when one of the hydrogen atoms separates from an OH group or hydroxyl group and joins with another one. And this becomes a water molecule. Then the remaining oxygen atom joins the two monomers, those two monosaccharides, They join those two together. Since a water is removed, we call this process, this reaction, a dehydration reaction. The opposite of this reaction is hydrolysis, and this is where you would force a water molecule back into the bond to separate it. Interestingly, this is what our bodies do to be able to use the energy stored in the molecules. This is a catabolic process. The longer the chain of saccharides, the more energy that is stored in it, because there's more bonds that we can break. So polysaccharides are large molecules that may consist of as many as 10,000 monosaccharide units linked together. And these are your complex carbohydrates. Now, complex carbohydrates is more of a nutritional name. The scientific name is the polysaccharides. They don't activate our taste receptors for sweetness like the mono and disaccharides do. They don't fit or interact as well with our sweet receptors. They're much larger and haven't been broken down yet. I just have a couple of slides here. This just kind of shows you how these all fit together. On this slide, you have our simple carbohydrates, our monosaccharides, glucose, fructose, and galactose. And those combine to form disaccharides. So a sucrose is a glucose and a fructose joined. Maltose is a glucose or two glucose molecules. And then lactose, which you'll find in dairy, is a glucose and galactose joined together. And then we have our complex carbohydrates, our polysaccharides. So in plants, you have our starches. You have amylose or amylopectin. If you notice, the amylopectin is branched. Each one of those are a little saccharide molecule, but they're just the little hexagon shapes in the chain are branched. Fibers are indigestible by humans. You have soluble and insoluble. And you've noticed the structure, what that looks like. And then we have glycogen. And this is stored in our muscle and liver. And if you notice again, it has a branched shape. All right. So this week we are going to do experiment 2.2, and I just want to give the background information for experiment 2.2 on page 40 and give you an example of an introduction or purpose for your lab report. So that's what you'll see on the screen here. You can pause it and read through it and look at it and copy it down on your lab report. Again, here's the introduction again. A hypothesis, a sample hypothesis, a prediction. And then when you are taking your data and observations for this, make sure you record what color the iodine turned when it came in contact with the sample, or did it remain the same color? You should have something written down for each one of the samples in the experiment. When you are looking to do your analysis or discussion and analysis section, be sure to answer these questions. Was there a difference? Which one could you conclude were polysaccharides? Which ones contained starch? Read the paragraph after the experiment to give you a little bit of guidance on how to write your conclusion or what information to put in there. This gives an explanation of what was happening in the experiment. So some possible observations you could have had during the experiment are listed on the slide. And this concludes the lecture for the first part of Chapter 2.